Hello and welcome to another video in this lecture series of advanced biomaterials. So in our previous videos we have seen about polymeric nanoparticles. So in today's video we will be looking at lipid nanoparticles or solid lipid nanoparticles and liposomes. What they are, their characteristics and some of their important applications and why they are important in different fields of medical engineering and designing novel therapeutic interventions for treatment of different diseases, in particular treatment of the in delivery of the vaccine for this novel coronavirus or COVID-19. So, lipid nanoparticles. So, what they are? Nanoparticles, these lipid nanoparticles are the nanoparticles which are composed of lipids. So, these are novel pharmaceutical drug delivery systems. So, lipid nanoparticles became very widely known in the late 2020s because some of the COVID-19 vaccines that use mRNA vaccines technology coat the fragile mRNA strands with polyethylene glycosylated lipid nanoparticles as their delivery vehicle. Now, these lipid nanoparticles can range in size from 10 to 1000 nanometers. So they have a wide size range distribution, and these are widely being known in the late 2020s because of the delivery of COVID-19 vaccines, the RNA of the COVID-19 vaccines as developed by Pfizer BioNTech COVID-19 vaccines by Pfizer. So these uses lipid nanoparticles. So in lipid nanoparticles or the solid lipid nanoparticles, there is only one phospholipid bilayer because the bulk of the interior of the particle is composed of lipophilic substances. So in solid lipid nanoparticles there is only one phospholipid bilayer, phospholipid layer. So if these are the phospholipid layers, bilayers, in lipid nanoparticles there will be only one such layer. In contrast, these liposomes are hollow lipid nanoparticles which have phospholipid bilayer as a coat because of these because of the bulk of the interior of the particle is composed of aqueous substance. So now coming to liposomes are another class. So these will be actually in form of a bilayeric system with a hollow interior core. So these liposomes will be having a hollow interior core, whereas the solid lipid nanoparticles will have a solid core. So these solid lipid nanoparticles are sphericals with an average diameter of 10 to 1000 nanometers. And these solid lipid nanoparticles contain a solid lipid core matrix that can stabilize lipophilic molecules. So this core substance part will have lipophilic molecules. Lipophilic molecules such as the different type of drug molecules will be lipophilic in nature. Now this lipid core will be stabilized by surfactants or emulsifiers. So this core will be stabilized by surfactants. These made up of? So these lipids if we consider them, so these can be composed of the mono, di and triglycerides, fatty acids, sterols such as cholesterol and also the waxes. So these can be made up of the composed of mono, di or triglycerides, then there can be the fatty acids, the steroids such as cholesterol, which is widely used for manufacturing the solid lipid nanoparticles, and the waxes. So now this was the general overview of lipid nanoparticles or solid lipid nanoparticles. So now we will look at the structure of liposomes. So erase out this part. So these liposomes now, these liposomes are spherical vehicles having at least one lipid bilayer. So these liposomes can have more than one lipid bilayer, but they need to have at least one lipid bilayer. These can also be used as drug delivery vehicles for administration of nutrients and different pharmaceutical drugs. But differing from the solid lipid nanoparticles or the lipid nanoparticles, these liposomes will be in the range of few micrometers to even tens of micrometers. So these are much larger than the lipid nanoparticles. And these liposomes can be produced by disrupting biological membranes such as sonication. So these can be produced through the disruption of biological membranes such as by ultrasonication. Now coming to the structure of the liposomes. So this outermost phospholipid bilayeric structure, these can be in multiple layers. So this part is the hydrophobic part. Hydrophobic part is the lipid loving or water repelling, and it will exist in this structure in an aqueous environment. So the outside part will be an aqueous environment, and also the interior part will also be an aqueous environment. So the advantages of liposomes is that these liposomes can be prepared for the delivery of both hydrophilic and as well as hydrophobic drugs. So the hydrophilic drug will be located in the inside space of the liposomes, whereas the hydrophobic drugs can be attached to the surface of the liposomes. So these liposomes are mainly composed of phospholipids such as phosphatidylcholine. Now, these liposomes has an aqueous solution core surrounded by this hydrophobic membrane in the form of a lipid bilayer. And the hydrophilic solutes are easily dissolved, as I mentioned, in the hydrophilic or in the core, and cannot easily pass through the bilayer. Now, just like we have discussed about the entry of polymeric nanoparticles in the cells, so these liposomes can also be complexed with certain drugs or certain DNA-based targets, therapeutic agents inside them through specific interactions, which are in this case known as lipoplex. So liposomes plus DNA will give us these lipoplexes. Now, unlike the entry of polymeric nanoparticles into the cells, which takes place through the process of endocytosis, these lipoplexes can enter the cells through membrane fusion or flip-flop processes, because the plasma membrane is also a lipid bilayer, and the membranes of these, the outermost membranes of both of these structures are, since they are similar, if they come into contact with such systems, then membrane will fuse, and the delivery of the aqueous of the drugs or the material which is present in the aqueous core of the liposomes will get inside the cells, and they will be transported to the desired site of action inside the cell. So this was the summary of the lipid nanoparticles and liposomes, which are one of the more recent approaches for different therapeutic interventions and drug delivery applications. So their preparation strategies are based on their applications or their construction or their the target or the active ingredient, biological ingredient for which they are being synthesized. So in our upcoming videos, we can look into some of the preparation strategies for these lipid nanoparticles or liposomes. So that will be all for today's video. Hope you enjoyed it. Thank you.